biosynthesis of these nucleotides. And then we also look at the degradation or the breakdown of these nucleotides. And then we will look at a few disorders that are related to the metabolic disorders of these nucleotides. So that's what we are going to do. So hopefully within the next 1 hour, 30 minutes, I think we should be able to go through all of these. And if there are any questions, contributions, we can have them. Right. So first of all, the biosynthesis of the nucleotides, or specifically the nitrogenous bases, we have different biomolecules, some amino acids and some other components. We'll look at them all that are involved in the biosynthesis of these nitrogenous bases. Now, I want you to remind yourselves of the fact that the nitrogenous bases are divided into two major groups, which are the purines and the pyrimidines. And the purines are adenine and guanine, and adenine and guanine are those that have two rings fused together, one 6-sided ring and one 5-sided ring fused together. And you have the pyrimidine, which is having a single 6-sided ring. So we'll first take the purines and look at their metabolism. Then we'll take the pyrimidine and look at their metabolism, which have similarities anyway, except that the sources of the components of their rings is different. The sources are different. So quickly we see that N1 in the purine ring is contributed by aspartic acid. That's an amino acid, aspartate. C2 and C8 come from tetrahydrofolate. That is what we commonly refer to one of these folic acids. We saw it earlier when we were treating coenzymes. So we saw folic acid as one of these components which is needed in the proliferation of cells and the development of neural tubes or the brain in fetal development. So we talked about that. Then we have N3, N9, contributed by glutamine, another amino acid. Then we have N7, C4 and C5, contributed by glycine. That's another amino acid. C6 is directly contributed by carbon dioxide. So you remember the last time I mentioned the fact that when we talk about respiration, we talk of it that as if when we take in air, the body uses the oxygen in metabolism and gives out the carbon dioxide. But really, that is not what happens, because you can actually see that carbon dioxide also has some use in the body. In fact, most of the carbon dioxide that we exhale out of the body comes from the metabolism, the metabolic processes that the cells undergo. So it is the carbon dioxide which is synthesized by the body that is actually taken out of the body in exhaled air. But the carbon dioxide, which comes with the air we breathe in from the atmosphere, most of it goes into these metabolic processes. So it has this utility, it has this utilization into different metabolic processes. So we can now appreciate the fact that carbon dioxide also has this importance in the involvement of metabolic processes in the body. Now, we take the biosynthesis of the purine ribonucleotides. So again, you know that we have the ribonucleotides and we have the deoxyribonucleotides. And the ribonucleotides are the nucleotides that form the ribonucleic acid, RNA. The deoxyribonucleotides are those that go into the synthesis of deoxyribonucleic acid, DNA. Now we are looking at the biosynthesis of the ribonucleotides. These are the ones that will go into the synthesis of RNA. And then we'll also look at how the deoxyribonucleotides are also synthesized. Now, the synthesis of the purine nucleotides is not separate on its own, but this synthesis occurs on the 5-carbon sugar, meaning that the synthesis of the purines is not done separately before the purine come to bind to the ribose, the 5-carbon sugar, but the synthesis is done on the sugar. I mean, most of the time. So actually, we'll also see the type of the synthesis where that synthesis is not de novo synthesis. It's not a synthesis that is that is coming from smaller molecules coming together, as we are seeing here. Here we see that smaller molecules, we take some amino acids, where we see folic acid coming together and then forming the rings all the way until we have a parent molecule, which is inosine monophosphate, from which other purine nucleotides are formed. But in the recycling of these nucleotides, then you can have, say, a separate nitrogenous base that will then come to bind to a sugar, which we will see later. But this particular one that we are seeing, that this is the de novo synthesis. De novo synthesis, meaning that the synthesis of the ribonucleotides are occurring from smaller molecules, step by step, coming together to form the nitrogenous base directly on the sugar. So let's go through the stages by which this is done. So the very first thing you see at the top is the ribose 5-phosphate, the very first molecule. Where do you get the ribose 5-phosphate from? Remember when we were doing metabolism of carbohydrates, one of the pathways that we briefly looked at was the pentose phosphate pathway, which is also known as the hexose monophosphate shunt. In that pathway, we saw the synthesis of ribose 5-phosphate, which then goes down to form ribose phosphate, and then into the synthesis of nucleic acids and coenzymes. So that ribose 5-phosphate, the very first thing which happens in the process of the synthesis of nucleotides is that it gains 2 phosphate groups from ATP. So we are seeing this, phosphorylation that we have been seeing with the various biochemical pathways. We are seeing it again here. This time around, it's a pyrophosphate, a diphosphate, 2 phosphate groups from ATP that gets conjugated on the ribose 5-phosphate to give you the phosphoribopyrophosphate. So the first molecule is having just 1 phosphate group on carbon number 5. But the product is having 2 phosphate groups in addition to that phosphate group, which is already on carbon number 5. We are seeing again 2 phosphate groups on carbon number 1. And those 2 phosphate groups are coming from ATP, cellular energy. You pay attention to the enzyme, which is also participating in that reaction, catalyzing that reaction. The phosphoribosylpyrophosphate pyrophosphate synthetase. So the naming, again, if you go back to our fundamentals in enzymology, that naming is based on the substrate on which the enzyme is acting. Next, now we see glutamine, one of the amino acids that we saw participating in the synthesis of nucleotides coming in. Now, glutamine then now comes to donate an amide group. That's an amino group that it comes to donate with participation of water. So you can look at it as, first of all, there is hydrolysis of the 2 phosphate groups from carbon 1, and then the amine group comes to replace the 2 phosphate groups. So you have a phosphoribosyl amine. So in place of these 2 phosphate groups on carbon number 1, we now have an amine group. Next, we see the second amino acid, glycine, the smallest amino acid. Also, with the participation of ATP, now comes to bind on the amine group, which was first donated by glutamine. So you now have a glycinamide ribosyl 5-phosphate. The process continues where now the folic acid, the tetrahydrofolate, comes into the reaction. So from here on, what you see is that because the formation of the ring is our focus, the formation of the purine ring is our focus. As it is getting more complex, we have reduced the sugar phosphate into just the writing we have below here, which is ribose 5-phosphate. We have written ribose 5P, that's ribose 5-phosphate. And then you are seeing that the purine ring, which is forming, is shown. That is our focus. That's what we are looking at. So after glycine, so you can pay attention even to the colors. Glutamine is red, so the group which is in the ring, which is red, that's coming from glutamine. And then we also see glycine is blue, and so that's glycine molecule that is binding onto the amine group, that's was first conjugated on the sugar is shown in blue. So the binding, that bonding is happening on the carboxylic acid group of the glycine which is coming in. So this one clearly shows that that's a peptide bond which is forming between the amine group that glutamine brought and then the carboxylic acid group of glycine, except that this is the residue which is binding to glycine. There is not it's not a protein, so you can really not refer to it so much as a peptide bond. Maybe you can say a peptide-like bond, which is between that amino group and glycine. The tetrahydrofolate is black, so we use black to represent that, and that's gives out that carbon with the oxygen, so it's black, which is binding to that amine group of the glycine residue that is binding in that molecule. So then we now have a formyl glycinamide ribosulfite phosphate. Right. You continue and you see that from that molecule, the formyl glycinamide ribosulfite phosphate, glutamine comes in again. And this time around, glutamine now again donates. So if you look at the structure of glutamine is having two amine groups. Now, glutamine comes in again and gives out another amine group, which is also red. So the components that are coming from glutamine are red, components that are coming from glycine are blue, and the component that is coming from the tetrahydrofolate is black. ATP comes into the reaction again and then closes the ring, so that with the removal of that double bond O on the carbon, the ring closes. So you now have the 5-sided ring formed. The imidazole ring of the purine molecule is now formed. From there on, you see the continuation of the formation of the ring to have the 6-sided part of the ring. First of all, we have carbon dioxide now coming in to donate that carbon, which is also black, on carbon number 4 of the glycine residue, which was used. So you see that carboxylic acid group, which is coming from the carbon dioxide on C4. The enzyme there is carboxylase. Of course, that is based on the process, the addition of carboxyl group. Now, you can also see that aspartic acid comes in again, this time around, to bind with that carboxyl group, which came in from carbon dioxide. But from that, we see that there's a removal of fumarate. So in other words, it's just the removal of the carbon chains that are in aspartic acid, leaving the amine group. So fumarate. So if you take out the amine group from aspartic acid, what you have left is an intermediate of the TCA cycle, fumarate. So that fumarate is given out. And so that fumarate obviously will go into the TCA cycle. And you are left with the amine group bonded, on that carbon. And the tetrahydrofolate comes in again And this time around, coming to donate another carbon, which now binds onto one of these amine groups. And eventually, you have in the very first reaction where you have the enzyme cyclohydrolase acting, there is some dehydration, and then there is a closing of the ring so that you now have the full purine ring formed, the 6-sided ring with the 5-sided ring formed. And that parent molecule is inosine monophosphate. That it is from this molecule that you have the synthesis of adenosine monophosphate, guanosine monophosphate, and then further phosphorylation of them will give you that they are diphosphates and they are triphosphates. So the parent molecule for the formation of purine nucleotides is the inosine monophosphate. And the synthesis of that inosine monophosphate is done with the participation of amino acids, 3 of them, aspartic acid, glutamine, and glycine, in addition to tetrahydrofolate. That's folic acid. So here again, you see the use of folic acid in the synthesis of nucleotides, which will go into the synthesis of nucleic acids. For the ribose 5-phosphate I mentioned, it is coming from the hexose monophosphate shunt or the pentose phosphate pathway, one of the pathways we saw in carbohydrate metabolism. So these are the things that you should pay attention to. Not so much of the specific reactions which we are looking at. So that even if those are difficult, not because you are not doing biochemistry major, this is just a course you are doing. I think the things that you should pay attention to is, first of all, what is the parent molecule, the product, that first product that comes from the synthesis of the purine ring, and that is inosine monophosphate. Then the sugar on which the synthesis of the purine ring is formed, the ribose 5-phosphate. Where is the sugar coming from? Then you can tell that that sugar is coming from the hexose monophosphate shunt or the pentose phosphate pathway. Then, what are the molecules that participate in the synthesis of the purine nucleotides? Glutamine, glycine, aspartic acid. These 3 amino acids, plus the formyl tetrahydrofolate, which we commonly refer to as folic acid. So 4 molecules that are involved, 4 compounds. 3 of them are amino acids, 1 folic acid, which come together to form the purine ring. So these are the things that you should pay attention to. You can have interest also in the enzymes that participate in the various reactions, like phosphoribosol pyrophosphate synthesis, formylotransferase, cyclohydrolase, which is responsible for the closing of the final ring, can pay attention to these enzymes as well. The reactions themselves, those of you who don't have so much of difficulty, and you can just look at the colors of the names of these compounds that are participating in the synthesis and correspond to the parts of the ring that are formed with those colors, and tell that this component is coming from glycine, this component is coming from glutamine, this component is coming from aspartic acid, this other one is coming from tetrahydrofolate. So you can appreciate the parts of the purine ring that are given by each of these molecules that are participating in the synthesis. Now, we have the inosine monophosphate, which we saw as the parent molecule, from which you then have the purine nucleotides that go into the synthesis of the nucleic acids, which means the inosine monophosphate will take two parts. One path should give us adenosine monophosphate. The second path should give us guanosine monophosphate. Now, the first path has the participation again of aspartic acid. And we see that this carbon with the cyclic ketone group on it, you have the aspartic acid coming to replace that oxygen. And then again, you have the removal of the carbon chain, which is in that molecule of amino acid, which is fumarate. In the end, you have an amine group that is replacing that oxygen that is on the original molecule of inosine monophosphate. That oxygen there is then replaced with an amine group. And remember, all of these is occurring on the ribose 5-phosphate sugar. So the ribose 5-phosphate sugar is already bonded to this, so that it's just a replacement of the oxygen with an amine group, which then gives you adenosine monophosphate. And that can into the synthesis of nucleic acids, specifically RNA. From this phosphorylation of it will give you can give you a triphosphate, which will have different functions than them participating in the synthesis of RNA. Now, the second path of inosine monophosphate first has some oxidation reaction with the participation of water. And you see that is bonded onto that 6-sided ring to give you xanthosine monophosphate. Then glutamine comes in and donates its amine group, which replaces that oxygen that came from water. That carbon which was oxidized, you now have an amine group from glutamine coming to replace that oxygen. And then you will now have the product guanosine monophosphate. That guanosine monophosphate can then go into the synthesis of RNA. If it undergoes further phosphorylation to give you guanosine diphosphate, guanosine triphosphate, they will have different functions. GTP will be the guanosine triphosphate is also a form of cellular energy, just like ATP. Now, we see that again, the amino acids that are involved here are similar amino acids which we have seen earlier. The aspartic acid, glutamine. Here we don't see tetrahydrofolate placing any role in terms of donation of parts. It is just addition of some, call it, if you look at it critically, it is just an attachment of amine groups coming from these amino acids to give you the guanosine monophosphate. So for instance, if you look at adenosine monophosphate, you see it's just an amine group which comes to replace that oxygen, and then the molecule is now complete, having given you the adenosine monophosphate. On the other part of the guanosine monophosphate formation, the amino acid that gives out the amine group is glutamine. So you can notice these two differences. Aspartic acid is what gives out the amine group in the synthesis of adenosine monophosphate from the parent molecule, inosine monophosphate, whilst in the synthesis of guanosine monophosphate from the parent molecule, inosine monophosphate, the amino acid that gives out the amine group there is glutamine. So you can notice these two differences. So as I was saying earlier, if you have that adenosine monophosphate and guanosine monophosphate, and you have a phosphorylation of these molecules to give you their diphosphates, those diphosphates can be phosphorylated further to give you their triphosphate, adenosine triphosphate, or guanosine triphosphate. Again, you see the enzymes that are catalyzing these reactions are kinases. So we keep saying that that kinases are enzymes that are involved in the transfer of phosphate group from one molecule onto the other. Here we see a nucleoside monophosphate kinase, nucleoside diphosphate kinase in these two reactions, being the enzymes catalyzing the transfer of phosphate groups from ATP onto these substrates to form their corresponding diphosphates and triphosphates. Now, let's talk about some health implications of this process of purine synthesis that we have just looked at. Now, so we want to look at application of this knowledge in the development of some antibiotics which fall under the group of sulfonamides. So we see the structure of sulfonamides, under which we have a sulfanylamide. Now, so sulfanylamide, one of the sulfonamides. This, so I mean, sulfanilamide sometimes is used as a synonym to sulfonamides. And looking at the structure, you see that it is a structural analog of para-aminobenzoic acid. Para-aminobenzoic acid. So the only difference you see between these two molecules, the sulfanilamide and para-aminobenzoic acid, is that in the sulfanilamide, you are having some sulfoamide group, which in place of that, you have a carboxylic acid group in the para-aminobenzoic acid. Now, in bacteria, we have the synthesis of the tetrahydrofolic acid or tetrahydrofolate occurring in the bacterial cell. So it means that tetrahydrofolate is synthesized in the bacterial cell. Now, we humans, we do not synthesize folic acid. We take in folic acid through food, or maybe sometimes through supplements. So the folic acid which is required by our cells, we take it in from food. But bacteria is able to synthesize these folic acid. And we have seen already that folic acid is needed for cell proliferation, cell development. And that is the reason why for pregnant women, folic acid is given to supplements in order to, if some deficiency is noticed, so that it will help with especially the development of the brain of the fetus. Now, if we have the process of synthesis of the folic acid, which we see on the right-hand side below, that we have the para-aminobenzoic acid that is reacting with dihydroterate diphosphate. And the reaction continues to the end where you have the formation of the tetrahydrofolic acid. If you have the para-aminobenzoic acid replaced with sulfanylamide, say if sulfanoamides, that group of antibiotics, are introduced into the system, what the antibiotic does is that it acts as a competitive inhibitor to the para-aminobenzoic acid. So again, the sulfanoamide and para-aminobenzoic acid are structural analogs. They look the same in structure, but functionally they are not the same. So if the sulfanoamide is introduced into the body and it goes to compete with the para-aminobenzoic acid, where the enzyme which is involved in that first reaction, the dihydroterate synthetase, now binds to the sulfanoamide, it prevents the reaction from continuing further. So that is competitive inhibition. We saw this when we were doing fundamentals of enzymology, competitive inhibition. So the sulfanoamide is acting as a competitor to the para-aminobenzoic acid. So once it goes to bind to the enzyme, it inhibits that reaction. It terminates the reaction. And so there will not be the formation of tetrahydrofollates for the bacteria. And that eventually will result in the death of the bacteria. First, it will result in inhibition of growth and proliferation of the bacterial cells. So they are not going to grow further. They are not going to multiply further. And then eventually, the bacteria dies as well. So that is an example of the use of the knowledge of enzymes, at the same time the synthesis of nucleic acids together, knowing the enzymes that that participate in these reactions, and at the same time there's the substrates, so that if you know the structure of the substrate and you introduce one which is a structural analog of the other, it goes to serve as an inhibitor to the process which you want to stop from occurring. We have another structural analog, methotrexate, which is a drug widely used in the treatment of some cancers. And methotrexate is a structural analog of folic acid. So which will mean that the folic acid itself, which is required acting as a coenzyme in various reactions in the proliferation of cells. If you have this drug, methotrexate, a structural analog of the tetrahydrofolic acid, it goes to inhibit the reactions that will result into the proliferation of cancer cells, so that you control the growth of those cancer cells. But of course, you have side effects of chemotherapy, which is to do with the fact that sometimes you have the drugs not affecting only the cancer cells, but they go to affect also normal cells. So that then results into some conditions of anemia, the loss of hair, some disorders of the skin, like some scaly skins, because it goes to affect those normal cells as well. So you see these as side effects occurring from chemotherapy. Right. Now, the synthesis of the purines we saw earlier was happening directly on the ribose sugar. I made that clear. And I also said you can have a path which is a recycling path when there is a degradation of nucleotide, where you now have a separate ribose sugar, separate nitrogenous base, and even you can have the phosphate group also separate. These ones can just come together and form a nucleotide. That is not the novo synthesis. It is not synthesis of the nucleotide from smaller molecules, from scratch. But it's just a recycling path. When there is a breakdown of these nucleotides, these components can bind together and then give you another nucleotide. In fact, this pathway, which is referred to as the salvage pathway, this recycling path will occur in some specific cells like erythrocytes, the red blood cells, some leukocytes, some white blood cells, and also in some neurons, cells of the brain. These cells do not have the process of the de novo synthesis of purines, or the novo synthesis of nucleotides occurring in them. Remember, I mean, these cells do not have a nucleus. I mean, for instance, the red blood cell, even though when you look at the stages of development of the red blood cell at younger stages of development of the red blood cell, there is some nuclear material, but by the time the cell develops fully to a mature red blood cell, that nuclear material is lost. So that you don't have a nucleus in the red blood cell. I mean, that is logically to give more space to hemoglobin so that the cell can carry out optimally its function of transport of oxygen and also carbon dioxide out of the body. Now, you see, so in those cells, you have this salvage pathway, which can occur, where you see, this is, so you see adenine, which is the nitrogenous base, and just with the participation of the enzyme adenine phosphoribosyltransferase transferase, you have that nitrogenous base binding onto the ribose 5 sugar to give you the adenosine monophosphate. Similar reaction occurs with guanine with the participation of the enzyme hypoxanthine guanine phosphoribosyltransferase transferase to give you the guanosine monophosphate. And then you also have another one, which is hypoxanthine. This is not a nucleotide which is involved in the synthesis of the nucleic acid, RNA or DNA, but it is to form that parent molecule, inosine monophosphate, from which you can have the synthesis of adenosine monophosphate and guanosine monophosphate. So you have these similar reactions of the formation of these purine nucleotides, the salvage pathway. Right. Now, you know, every biochemical reaction has some form of regulation. There should be a coordination of these reactions such that the products are in adequate amounts for the cells at all times to carry out their functions. Now, when you look at this process, which we looked at earlier, the biosynthesis of the purine nucleotide, you realize that first of all, for these reactions to occur, you need the ribose 5 phosphate, the sugar, that 5 carbon sugar, which is produced from the pentose phosphate pathway, is needed, first of all, for the reaction to occur. Then you also need at least the very first enzyme, the phosphoribosyl pyrophosphate synthetase, that is involved in that first phosphorylation reaction to give you the phosphoribosyl pyrophosphate. You need these two for the initiation of the reaction to occur. Now, to some extent, you can say that you also need energy, which is ATP, which is giving us the very first, phosphorylation to occur, the two phosphate groups that come to bind to that ribose 5-phosphate to give you its pyrophosphate. So at least you need three things for the reaction to occur. First, the sugar, the sugar, the ribose 5-phosphate. You need the enzyme, phosphoribosylate pyrophosphate, that will transfer the two phosphate groups from ATP, which means you need ATP. And then you need that enzyme. So these three things are needed for the reaction to get initiated. Now, in the regulation of the synthesis of the purine nucleotides, we have the concentration of the phosphoribosylate pyrophosphate playing a critical role, so that we need enough of this phosphoribosylate pyrophosphate in order for the first committed reaction of glutamine coming in to now give us the phosphoribosylate amine. That glutamine that comes into the reaction even before glycine comes, the tetrahydrofolate comes, and then the formation occurs. So we first need that pyrophosphate to be in adequate amounts in order to have adequate biosynthesis of the nucleotides, the purine nucleotides. So that is a factor that regulates the process of the biosynthesis of purine nucleotides. Now, other factors that are involved in what we refer to as feedback mechanism, which we have seen much earlier. We saw feedback mechanism where products of a reaction now serve as regulators to enzymes in the process. Here we have the adenosine monophosphate and guanosine monophosphate that act as inhibitors to enzyme of the second reaction in the process, which is the phosphoribosylate pyrophosphate glutamyl amidinotransferase. The first enzyme which is involved in the transfer of the amine group from glutamine. So the regulation or the control of the process of the biosynthesis of these purine nucleotides is occurring from the regulation of the activity of that enzyme, so that higher concentration of adenosine monophosphate, higher concentration of guanosine monophosphate act as inhibitors to that process. What it means is that less of adenosine triphosphate is going to inhibit, because it means that much of the adenosine triphosphate had been converted, like they had been used, so that there isn't enough energy for the first reaction to occur, because you need an ATP from which the two phosphate groups will bind onto the phosphoribosylate to give you its phosphoribosylate before the glutamine comes in. So if you are having a lot of the monophosphates and have less of ATP, the triphosphate, then the reaction shouldn't go on, making use of more energy. And so that is why you see the adenosine monophosphate and guanosine monophosphate inhibiting that that enzyme, that glutamyl aminotransferase, which is involved in the transfer of the amine group from glutamine in the process of that biosynthesis of nucleotides. Right. Now, you also have after the synthesis of inosine monophosphate, the inosine monophosphate goes into two different paths, which we saw. One of the parts is going to give us adenosine monophosphate, AMP, and the second part, gave us guanosine monophosphate, GMP. Now, these two final products, AMP and GMP, also serve as regulators of the synthesis of these nucleotides. First of all, the adenosine monophosphate serves as an inhibitor to the enzyme of the first reaction where aspartic acid comes in to give its amine group onto the molecule with the removal of fumarate. So that adenosine monophosphate, the product in the process, serves as an inhibitor to adenyl succinate synthetase, that first enzyme involved in the conjugation of aspartic acid to inosine monophosphate to form adenyl succinate. Similarly, guanosine monophosphate, GMP, serves as an inhibitor to the first enzyme, inosine monophosphate dehydrogenase, which is involved in the oxidation of inosine monophosphate to form xanthosine monophosphate, where you see the oxygen from water getting conjugated onto the inosine monophosphate. So we see that form of regulation also occurring. Adenosine monophosphate serving as the inhibitor to the first enzyme in the conversion of the inosine monophosphate. And we see guanosine monophosphate acting as an inhibitor also to the first enzyme, inosine monophosphate dehydrogenase. Okay. Now, so far, we have been talking about ribonucleotides. But we also have deoxyribonucleotides. The ribonucleotides are those that go into the synthesis of RNA, whilst we have the deoxyribonucleotides going into the synthesis of DNA. But so far, we've talked about only the ribonucleotides. Now, how do we get the deoxyribonucleotides, which will go into the synthesis of DNA? Now, the ribonucleotides that we have synthesized is having the sugar ribose. And just by reduction of the group, the hydroxyl group, which is on carbon number 2, so that in place of that hydroxyl group, we will have only hydrogen that gives us a deoxyribonucleoside. And in that process, we have the enzyme ribonucleotide reductase with some removal of water. Now, the enzyme ribonucleotide reductase itself, will be acting together with thioredoxin, which after the ribonucleotide reductase actually directly gives out hydride ions onto the ribonucleoside, the diphosphate, to give us, to replace the hydroxyl group with that hydrogen. And once it does that, there will be the need to have that ribonucleotide reductase reduced back so that it can continue the reaction. And that is where thioredoxin comes in that reaction to give out the hydrogen or the hydrides or the protons back onto the enzyme ribonucleotide reductase. And then itself will have to get reduced again by thioredoxin reductase with the participation of the coenzyme NADPH, which also we saw earlier when we were treating coenzymes as the coenzyme, which is involved mostly in reduction reactions. We saw the NAD plus as the nicotinamide dinucleotide, which is involved in oxidation reactions, and the NADPH to be its corresponding coenzyme involved in reduction reactions. Now, here again, an application. We have the drug hydroxyurea, which is also used in the treatment of cancers, serving as an inhibitor to the enzyme ribonucleotide reductase. In other words, that drug, the hydroxyurea, is preventing the formation of deoxyribonucleotides so that the cancer cells, once they have the synthesis of DNA in them being impeded, being impaired, they will not be transferring the information from the cancer cells to other cancer cells, so that you curtail the reproduction of the cancer cells. So that is another application of this knowledge in the treatment of cancers. Now, further, health implication we can refer to with the metabolism of nucleotides. This time with the degradation of nucleotides. So we have been looking at the biosynthesis, which is the anabolism of these purine nucleotides. Now we have moved to degradation, which is catabolism, the breakdown. Now, when you take the two purine nucleotides that we saw, first adenosine monophosphate, and then guanosine monophosphate. So each of them, the very first reaction which occurs in terms of their breakdown is that there is the removal of the phosphate group from them. That is dephosphorylation. And once the phosphate group is removed from adenosine monophosphate, you now have just adenosine, which means the base bound on the ribose, no phosphate group. The same thing to do with guanosine monophosphate. Dephosphorylation occurs and you are left with guanosine, So from guanosine monophosphate, you now have just guanosine, just the base bounded with the ribose. Now, there is a third one here, which is the inosine monophosphate. Again, that inosine monophosphate is the parent molecule from which these nucleotides are formed. We saw already. And with the participation of adenosine monophosphate deaminase. So that is a deamination, just like that deamination we saw with amino acids. So the amine group, which is on adenosine, the six-sided ring is removed, and we have the formation of inosine monophosphates. Now you see that the dephosphorylation reactions are occurring under the activity of nucleotidases. These are the enzymes that are involved in the removal of the phosphate group. So you see that removal of phosphate group from adenosine monophosphate, the enzyme is the nucleotidase. Removal of phosphate group from inosine monophosphate, IMP, is catalyzed by, again, the nucleotidase. Similarly, from the guanosine monophosphate is also a nucleotidase. Then we have deamination, from either the adenosine monophosphate or just adenosine. We have the enzymes that are catalyzing those reactions being deaminases. If it is from adenosine, it is adenosine deaminase. If it is from adenosine monophosphate, then it is the adenosine monophosphate deaminase. Now we see that if you take the path of adenosine monophosphate, that is undergoing first dephosphorylation and deamination, the product it gives us is the inosine. And so that will be inosinic acid that is bounded on the ribose sugar. If you look at it from the AMP first getting undergoing deamination. So it's just that the metabolism of the breakdown of adenosine monophosphate is taking two paths. It is either, first of all, there is dephosphorylation. Phosphate group is removed first of all to give you adenosine before deamination occurs to give you inosine. Or you have first deamination occurring before dephosphorylation. But these two paths will end up giving you inosine. You either remove the phosphate group first before you remove the amine group, or the amine group is removed first before the phosphate group is removed. Both of these paths end up giving you one product, which is inosine. And then from there on, you have further reactions where you have phosphorylation reaction, where inorganic phosphate group is bonded onto inosine to give you hypoxanthine. Then you now have xanthine oxidase coming in that reaction to give you xanthine. If you look at the guanosine monophosphate path, where you have first the dephosphorylation occurring, and then you have guanosine, and that guanosine then sort of after dephosphorylation, you now then have phosphorylation again to give you guanine. But this phosphorylation is occurring together with the removal of the ribose sugar at the same time, so that you are left with just the guanine and not the sugar. So the energy which comes in with that second phosphorylation goes to the removal of the sugar to be ribose 5-phosphate and not just ribose. We also see that similar reaction occurring with inosine, where you have the phosphate group coming in, and then there will be the removal of the ribose 1-phosphate, giving us hypoxanthine. But you see, these paths, all of these paths come to join at one product, which is xanthine. So the xanthine is that merging point of the breakdown of adenosine monophosphate, guanosine monophosphate, and even their parent molecule, which is the inosine monophosphate. They all merge, the degradation merges at xanthine. And then from xanthine, you have an oxidation reaction under xanthine oxidase. And the final product is uric acid. So you keep that in mind. In humans, uric acid is the final product of the degradation of purines. Uric acid is the final product of degradation of purines in humans. Now, in other animals, other than primates, like some fishes, for instance, you have uric acid undergoing further oxidation to give you a product, allantoin, which will then get excreted. In some others, some fishes, even amphibians, mollusks, you have that allantoin undergoing further reaction, say carboxylation, to now give you allantoic acid, which will get excreted. And the reaction can continue in other organisms like marine invertebrates, where you have even the allantoic acid getting converted to urea, to ammonia, and getting excreted. In amphibians and most fishes and mollusks, you have the allantoic acid after it gets it undergoes further reaction into the formation of urea and get excreted in those amphibians and fishes and mollusks as urea, whilst in ammonia, in marine invertebrates, you have the urea undergoing further reaction to form ammonia and getting excreted out of the body. So you see the end products in humans not the same as in other organisms. In humans, it ends at uric acid, but in other organisms, fishes, marine invertebrates, amphibians, you see different products that are excreted because of further reactions of the uric acid in those organisms. Now, these two has some health implications. Now, if you have uric acid, which is being produced in elevated amounts and found in such elevated amounts in blood serum, that can first of all, that condition is referred to as hyperuricemia, but the elevated amounts of this uric acid can go into the deposition of this uric acid as its salts, usually as sodium salts of the uric acid, which we refer to as sodium urate. And the deposition of these sodium salts of uric acid usually occurs in soft tissues, the cartilages in the joints, some occurring in the kidneys, and this can result, if it is in the kidneys, into what is into, say, kidney stones, what is referred to as kidney stones, or can result into some renal damage, or even kidney stones in the ureter, the tube that is connecting the kidneys to the bladder. The deposition of these sodium salts of uric acid in the soft tissues, that's the cartilages in the joints, will result in goutic arthritis, causing inflammation in the joints, which we can cause pain in the joints, a condition normally referred to as gout. You have also, now the reaction pathways which you see here, actually we have seen much earlier. I mean, this is just from glycogen or glucose. You have glucose 6-phosphate, and you have the pentose monophosphate chart, from which you have the ribose 5-phosphate. And then from there, you have glutamine and phosphoribosylpyrophosphate pyrophosphate, what we just saw, going down the path to give us the inosine monophosphate. And from it, you have the degradation giving us hypoxanthine. Now, you see, from hypoxanthine to the formation of uric acid, we have the xanthine oxidase involved in the oxidation of hypoxanthine to xanthine, and then xanthine to uric acid. Now, the lack of feedback inhibition, which we saw earlier with the pyrophosphates, that phosphoribosyl pyrophosphate synthesis. We saw the diphosphates, ADP, GDP, and also the glutamyl transferase. So the glutamyl transferase, which is involved in the transfer of the amine group from glutamine onto the phosphoribosyl pyrophosphate. That enzyme is one that was involved in the regulation of the process, so that the lack of feedback to this enzyme will result in excessive production of uric acid, because then once that enzyme is not regulated, it means the reaction will continue to be forming more inosine monophosphates, which will then undergo more degradation, and then will result into the formation of uric acid in large amounts. But we have the drug allopurinol, which is also an inhibitor to xanthine oxidase. So that is a drug which is used for the management of hyperuricemia and goutic arthritis, where the allopurinol acts as a structural analog. It's an inhibitor having similar structure but different functions to the xanthine oxidase, so that if it inhibits xanthine oxidase, there will be less formation of xanthine from hypoxanthine. Similarly, there is going to be less formation of uric acid from xanthine, so that the concentration of uric acid is lowered in that process. So that then can alleviate the accumulation of uric acid in the joints, which will be causing inflammation. That is the goutic arthritis, and also the formation of these kidney stones, both in the kidneys and in the ureter. You can have allopurinol being used for the treatment and management of these conditions. Now we look at a genetic condition referred to as the Lesch-Nyhan syndrome, which is also related to the metabolism of nucleic acids. So we saw the novo synthesis of the purines, resulting into the production of inosine monophosphate, and this inosine monophosphate goes into the synthesis of guanosine monophosphate, and then adenosine monophosphate. So adenosine monophosphate, guanosine monophosphate were produced from inosine monophosphate. And we have also seen the degradation of these nucleotides that were formed to guanosine and then guanine, and that guanine can break down, further to xanthine, and then from xanthine the uric acid is formed. Similarly, the adenosine monophosphate, the breakdown of it will give us adenosine, which goes into the formation of inosine, coming down to hypoxanthine and then xanthine, and all of it end up with the formation of uric acid, and which we saw the activity of xanthine oxidase in the formation of xanthine from hypoxanthine, and the formation of uric acid from xanthine. We saw all of that. Now there is an there is this inherited disorder which is to do with the gene which is responsible for the synthesis of the proteins that become the enzyme hypoxanthine phosphorybosyl transferase. The HPRT on the diagram is the same thing which is referred to as the HTPRT. Here in the gene is referring to guanine. So sometimes the enzyme is called as a hypoxanthine guanine phosphorybosol transferase, but sometimes the guanine is not referred to, but it's the same thing that is being referred to. So what you see, that enzyme, the hypoxanthine phosphorybosol transferase, or the hypoxanthine guanine phosphorybosol transferase, referring to the same thing, HPRT or HGPRT. The deficiency of it means that the breakdown of guanosine monophosphate, which eventually results in also the formation of a guanine, the HPRT is involved in the recycling of guanine to give us back the guanosine monophosphate. It is also involved in the recycling of hypoxanthine to give us back inosine monophosphate. Now, the deficiency of this enzyme will then result in accumulation of guanine, because you don't have this much of the enzyme which will be converting the guanine back to guanosine monophosphate. And so the other pathway for the guanine is to go into the synthesis of xanthine. So you see the guanine getting converted into more xanthine than going into the recycling part of the formation of guanosine monophosphate. Similarly, hypoxanthine with deficiency of the HPRT means that less of inosine monophosphate is going to be formed, and the other pathway left for the hypoxanthine will be to go into the formation of xanthine. So there's going to be an accumulation of xanthine, and that xanthine will then go into the formation of uric acid, and that will result in excessive uric acid being formed in the system. So you have excessive production of uric acid, which we saw earlier that can result into some nervous system impairment. But in this case, it's very severe. It's a very severe condition that occurs mostly in males, or let me say in males, because the genetic disorder that occurs is on the sex chromosomes. And for a female that is having an XX chromosomes, even if you have such a deficiency occurring, such a female will only be a carrier, and the result of it is asymptomatic. But in males, it's severe, such that such persons that have the deficiency of this enzyme and having such an accumulation of uric acid in their systems, which will be having nervous system impairment, resulting into mental retardation and some aggressive behavior. And also you have in such persons having an urge, grieving, just biting their fingers. So many of these, they will bite their fingers to bleed, and they continue to bite. And even in some very, very severe cases, some people even injure their tongues, they chew even their tongues and all of that. So you have that condition as the Lesch-Nyhan syndrome, also related to the metabolism of these purine nucleotides. Now the pyrimidines. So just as we saw different compounds which were donating different parts of the purine rings, here we also see two amino acids, glutamine and aspartic acid, which are donating different parts of the pyrimidine ring. We also see one of the carbons being contributed directly by carbon dioxide. In the formation of the purine ring, we saw the carbons were being donated by the tetrahydrofolate. Here we are seeing carbon dioxide donating that. I think with the formation of the purine ring, yes, there was carbon dioxide that was donating one of the carbons, and the rest of the carbons were donated by the tetrahydrofolate. Here again, we see carbon dioxide contributing one carbon in that ring, and the rest of the parts are contributed by amino acids, glutamine, and aspartic acid or aspartate. And then again, so the difference between the synthesis of the pyrimidine ring and the purine ring is that the purine ring, as you saw, was being synthesized directly on the ribose 5-phosphate to form the nucleotide. In the case of pyrimidines, the pyrimidine ring is synthesized separately before it gets conjugated onto the ribose 5-phosphate. So that is a major difference between the synthesis of purines and pyrimidines. Now, here too, if you look at the reaction pathway. We see carbon dioxide, glutamine, first of all, undergoing some conjugation with phosphorylation, because there is also the participation of ATP, donating one phosphate group in that process. And what you see between the carbon dioxide and glutamine and the phosphate group from ATP forming, the first molecule is carbamoyl phosphate. And then the reaction continues with aspartic acid coming in to contribute to the blue part. So the red part is coming from glutamine, which is just the amine group coming from glutamine. And the blue part is coming from aspartic acid, whilst the black C, that's the carbon, is coming from carbon dioxide. So this process is much easier, much shorter than what we saw with the synthesis of the purines. I mean, that is because the purine is having two rings. One, first of all, the imidazole ring, which is the 5-sided ring before the 6-sided ring. Here is only one 6-sided ring, and so it's easily formed. And you have from that carbamoyl aspartate dehydration process, under the activity of dihydroorotate, you have the closure of that ring. And then we have further processes of oxidation, which produces orotate or orotic acid in that process. Now, the process continues all the way with further, so you now have a conjugation of the phosphoribosylpyrophosphates coming in that reaction, and with a loss of a pyrophosphate or a diphosphate, you have a conjugation onto the sugar, the ribose 5-phosphate, to give you orotidine monophosphate, OMP. From there on, what you see is there is some decarboxylation, OMP decarboxylase, in that reaction, where there is a loss of the carboxyl group. That carboxyl group there is lost, and so you now have the ribose 5-phosphate bonded directly on that N1, the nitrogen one, to give you uridine monophosphate. Now you can say that the parent molecule which is formed in the synthesis of pyrimidines is uridine monophosphate. Just as you said, the parent molecule which is formed in purine metabolism is inosine monophosphate. Here it is uridine monophosphate, because it is from uridine monophosphate that you now have the synthesis of uridine diphosphates. You have so deoxyuridine monophosphate, deoxy uridine diphosphates, and then the deoxy thymidine monophosphate. You also have the uridine triphosphate, and then the cytosine triphosphate. So you have the synthesis of all these nucleotides from the three different nitrogenous bases, which are pyrimidines, which is cytosine, uracil, and thymine being formed in this pathway. Now, the regulation of this process is such that you have first of all, the enzyme, the first enzyme, the carbamoyl phosphate synthetase 2, is activated by the phosphoribosyl pyrophosphate. I mean, that is, first of all, one of the compounds that are needed for the synthesis of the pyrimidine nucleotides. As you can see from orotate or orotic acid, the continuation at the top there, you see the phosphorybosyl or pyrophosphate coming in, which means the concentration of it in the system regulates the synthesis of these pyrimidine nucleotides. It will have to be there in adequate amounts in order for it to bind to orotic acid for the synthesis of these pyrimidine ribonucleotides. But this enzyme, the carbamoyl phosphate synthetase 2, that first enzyme there, can also be regulated by feedback mechanism. And by that, the products, uridine diphosphate, UDP, and then uridine triphosphate, can inhibit the activity of that enzyme. Meaning when these two, the uridine diphosphate and uridine triphosphate, are produced in high amounts, they then give feedback to the enzyme so that there will not be further formation of carbamoyl phosphate. Similarly, you have the CTP, that is the citidine triphosphate, UTP, uridine triphosphate, also inhibiting the second enzyme, aspartate transcarbamoylase, which is involved in the conjugation of aspartic acid to carbamoyl phosphate. You also have the OMP decarboxylase, which is the enzyme involved in the decarboxylation of orotidine monophosphate. That enzyme can also be inhibited by products of the reaction pathway, which is uridine monophosphate. The first product that is formed when you have that OMP decarboxylase forming is uridine monophosphate, what I refer to as what you can refer to as the parent molecule, the uridine monophosphate. That together with CMP, which is citidine monophosphate, acting as inhibitors to OMP decarboxylase. So you have these different ways of regulation of the process of the biosynthesis of pyrimidine ribonucleotides. Now, so that was about synthesis of these pyrimidines. Now the degradation. So again, the degradation of these nucleotides is similar to what happens with the degradation of purine nucleotides. Same processes of dephosphorylation, deamination, and also the cleavage, the breakdown of the glycosidic bond, which is between the sugar and the nitrogenous base, so that you have the components separated. So similar processes occur with the breakdown of pyrimidine nucleotide, so that in the end, you will be left with just the cytosine, uracil, and thymine, the nitrogenous bases. The sugars will be separate, the phosphate group will be separate with the processes of dephosphorylation, deamination, and a breakdown of the glycosidic bonds. Then you can have further the bases. After the bases have been separated from the phosphate sugar, these bases can get degraded further to amino acids, beta alanine and beta amino isobutyrate. And these amino acids will then go into what we saw earlier, metabolism of amino acids. When we're looking at metabolism of proteins, this will then go into the processes of transamination and deamination, and will produce acetyl-CoA, succinyl-CoA, in which will go into the TCA cycle. So we are seeing the interconnectedness of these metabolic pathways. None of them is a standalone. In the end, you see the these nitrogenous bases breaking down into amino acids. Remember, these rings were formed with the participation of amino acids. So as they get broken down, they break down into amino acids, and then these amino acids then undergo processes of transamination, deamination, and then the carbon chain will go into the production of acetyl-CoA, succinyl-CoA, which will then go into the TCA cycle that we saw when we were dealing with the metabolism of carbohydrates. The salvage pathway of pyrimidines will also serve as, that recycling of the nucleotide, the nitrogenous bases into the formation of nucleotides, so that apart from what we saw, the de novo synthesis of pyrimidines, you have the nitrogenous bases, these pyrimidines, binding onto their respective phosphosugars to give you their respective nucleotides, just like what we saw in the salvage pathway of the purine nucleotides. Now here again, disorders of the pyrimidine metabolism are related to the accumulation of orotate or orotic acid, which condition we refer to as orotic aciduria. And of course, that also results in severe anemia and retarded growth, because it affects also the nervous system. And that can be caused by a deficiency of the enzyme, the orotate phosphoribosol transferase. So you see the first column, the reaction ended with orotate, which is the same as orotic acid. And then the reaction continues from that, the orotic acid or the orotate, the enzyme that acts on it to give you the orotidine monophosphate, OMP. That enzyme is orotate phosphoribosol transferase, which means that a deficiency of this enzyme will result in the accumulation of orotic acid, which then, will result into orotic aciduria, which causes, so there will be excretion of orotic acid in high amounts in urine, and also that can result into some damage of the nervous system. Other symptoms include severe anemia. Of course, the damage of the nervous system will result into some retarded growth and some nervous system disorders. We also have what we refer to as the Ray's syndrome. The Ray's syndrome actually is also it's just similar to the orotic aciduria, but it's referred to as a secondary orotic aciduria, because it is not coming directly from what we saw earlier with the deficiency in that enzyme, the orotate phosphoribosol transferase, but rather you have this occurring from a defect in another enzyme, ornithine transcarbamylase, which is involved in the urea cycle. I mean, that is another cycle that we don't have the scope of this course covering. Of course, if you are interested, you can look briefly at it. You see that the metabolism of these pyrimidines, amino acids, have a connection with the urea cycle, where you have the formation of urea. And because you have a defect in that enzyme in the urea cycle, which results in the accumulation of carbamoyl phosphate. So we see the carbamoyl phosphate is the very first product which is formed when you have the carbon dioxide and glutamine reacting. The very first reaction, the product that you see there is carbamoyl phosphate. So that you have a defect in the enzyme ornithine transcarbamylase, resulting in the accumulation of that carbamoyl phosphate. And so that carbamoyl phosphate, once there's accumulation of it, it is diverted for increased synthesis and excretion of orotic acid. Of course, once you have in large amounts carbamoyl phosphate, it means the reaction part it will continue so that it forms that orotate or orotic acid, which means an accumulation of carbamoyl phosphate results in the overproduction of orotic acid. And the overproduction of orotic acid we have seen will be causing orotic aciduria. So it's the same as orotic aciduria, but this time around, there's a connection with another enzyme, which is in a urea cycle. That's why the Ray's syndrome is referred to as a secondary orotic aciduria in that process. So this is where we will end the metabolism of nucleotides. From now, I think what we'll do is I will just open the floor now